Thank you very much, Hans. Thank you very much for the introduction. It's always a pleasure to participate in this webinar, and thank you to all of you who are listening now. I believe you can hear me well and you can see my presentation.
Yes.
Perfect. Thank you very much. So today we'll talk about the automated crystallography pipelines based on the CrystalDirect technology and the CRIM software that we have developed in Grenoble. But the motto of this iNEXT webinar series is, well, what can iNEXT do for you? To answer the question in a nutshell, that's the take-home message. In Grenoble, through iNEXT, we offer access to 3 different crystallography pipelines: a pipeline for protein to structure determination for new structures, a pipeline for compound screening, and for example, in the case you have identified a series of small molecules binding to your targets that you can characterize structurally, and a fragment library screening pipeline that allows you to do large-scale, fully automated fragment screening. All these pipelines are fully automated. They are remote controlled over the internet, and they are based on these unique technologies, CrystalDirect and CRIMS, that I will be introducing today. So if you are interested, you can have funded access to these pipelines through the iNEXT project. In this talk, I will first introduce the H6 lab in Grenoble, then present the CrystalDirect and CRIMS technologies, and then I will describe how we have connected these technologies to offer these crystallography pipelines that are services that are available to you in Grenoble. If I have time, I will also tell you about some new developments that can actually lead to new services for the rapid analysis of membrane proteins through serial crystallography, which, as you know, are very important targets in biology. But let's go to the first topic. The H6 lab, my lab in Grenoble, is at the EMBL site in Grenoble. As you know, EMBL and ESRF have a close collaboration, and EMBL and ESRF operate together the macromolecular crystallography beamlines at the synchrotron. And our team offers fully automated crystallization, but also crystallography pipelines to users worldwide. Since we started operations in this facility in 2003, we have supported more than nearly 1,000 users worldwide, and many of them are actually from external users that come from many countries. These users typically access either through easy-funded projects like iNEXT-Discovery in this case. Sometimes they are users from industry, in which case they access through proprietary access mechanisms, but both things are possible, and we have quite a considerable throughput yearly in the platform. But in addition to providing services, our team has a strong focus on technology development, and today we'll be introducing these 2 key technologies that we use to develop automated crystallography pipelines. As I said, the focus of this development is automation. And as you know, structural biologists have been investing in introducing automation at different levels of the crystallography pipeline. Today, crystallization, but also data collection, even data analysis happens in a very automated way and in very automated facilities. But for some time, there was a gap between these highly automated processes, which was the process of mounting crystals, extracting crystals from crystallization supports and preparing them for data collection. And our team, in collaboration with the team from Florence Cipriani, the instrumentation team in EMBL Grenoble, developed what we call the CrystalDirect concept that enables full automation of the crystal mounting and processing process. And with this, we were able to connect all these different processes to build fully automated crystallography pipelines that I will be describing today. But once we did this, it was obvious that having the hardware pipelines is not enough. If you want to use this pipeline efficiently, you have to invest a lot in software and data management that connects the samples along this process. And this is what we did. And we developed the crystallographic, information management system, CRIMS. That is a web-based system that allows you to design crystallization experiments and follow and track your samples along these pipelines automatically while the samples progress through the pipeline. And it's the combination of the hardware and the software that allow us to be so efficient in this process. With this, we established what we like to call online crystallography, which is a concept where you are controlling a crystallization facility and data collection beamlines at one or multiple synchrotrons from your computer. It's like if we put a crystallization facility and a synchrotron in your lab, accessible through your desktop. So, but how does automatic crystal harvesting work with CrystalDirect? Well, many people have tried to automate the process of crystal harvesting, and here you have some of the previous publications using joystick manipulated robots, optical tweezers, acoustic ejection, but to the best of our knowledge, no one has actually been able to fully automate the whole process in a satisfactory way. For this, we came up with a new concept that we call CrystalDirect, which consists on growing crystals on slightly modified vapor diffusion plates, where the crystallization drops are set up on thin films. And to extract the crystals, we simply use cut the film around the crystal and glue this piece of film containing the crystal to a standard data collection pin that we use at all synchrotrons. And of course, the most precise way to cut this film that we found and the most automated was to use a laser system. This is the CrystalDirect machine, as you could see it in the lab, in our lab in Grenoble. This is the CrystalDirect harvester. Here you have this very powerful laser beam. This is a samples change robot that does the opposite of what the robots do at the beamline. It takes frozen crystals and stores them in a cryogenic tank before we transfer them to the beamline, and this is the high dework capacity. In a detail, here we have the plate with the crystals, and there is an optical path. This is the laser here, and there is an optical path that, and this is the system that will steer the laser to do the excision during the mounting process. We have a sort of pins, standard data collection pins, a cryostream, and well, other components that are important. So how does the system work? Well, in CRIMS, the crystallographic information management system, you can recover images from your crystallization trace automatically through the web. And this is an example here. So I'm going to activate this video. When you have the images from your drops through the web, you can, I hope, okay,
yes.
So through this web interface, you can see your crystals, and you can use this these utilities on the right, select the crystal shapes that are compatible with your crystals. You can position, and this is going to determine how your crystals are going to be automatically harvested later. The system will record all the harvesting operations for your plates, and then we will move the plates. An operator in the lab will move the plate to the harvester. This typically happens offline, and then the system will automatically recover all the harvesting operations and will apply automatic harvesting one after another. Here you see how the harvesting operation starts by making an aperture on the film through which we remove the crystallization solution. Then the system is already putting a little bit of glue to the tip of the pin, and the system puts the pin in contact with the outside part of the film, and then a laser system cuts the shape around the crystals, and then the system recovers the pin with the crystals at its tip, transfer it to a cryostream, and then the sample change robot recovers the crystals from the cryostream and puts them in the storage. And even before this is finished, you see that the next harvesting operation for the next crystal is happening in a completely automated way. So you see it's a bit faster. I could hardly catch up. It's about 1.5 minutes per harvesting. So we have been using this technique in the lab for quite a long time now. We have harvested thousands of crystals, and obviously it has a number of advantages. First, it's fully automated and high throughput, but it also tends to represent less mechanical stress for the crystals because we don't come inside with tools. So fragile crystals tend to stand better with this during mounting with this technology. Very often we don't need to add cryoprotectants because of the way we process the samples. I don't have time to go into the details, but this is an advantage that you eliminate one variable from the equation. Nevertheless, if it is necessary to do it, we can add cryoprotectants as well, and we can mount multiple crystals in a single pin, which allow us to increase the density, the sample density. That could be interesting to increase efficiency at the synchrotrons. So what we did then was to combine the CrystalDirect technology with the high throughput crystal production at the facility and the automated data collection pipelines at the ESRF to provide fully automated pipelines. And this is a project that was driven by Irina Kornatsu, who is a staff scientist in my team and who did a fantastic work also in collaboration with the MX group from the ESRF, the instrumentation team, and the EMBL diffraction team. Now, one problem that we need to solve at that level was to improve communication between the home lab and the synchrotron. You know that samples are produced in the home lab, sent to a synchrotron, data is collected there, some parameters are stored there, data sets are typically replicated, but typically people use several synchrotrons. You may collaborate with other labs that will also produce samples, and at some point, this process can be quite complex. There is a lot of metadata involved in the process, and you know, doing this in high throughput can be a bit difficult, even in low throughput, I would say. Our answer to this was CRIMS, the Crystallographic Information Management System. As I say, that is able to manage not only follow the samples along the whole workflow, but also provides automated communications between the home lab and the synchrotrons. CRIMS was initially developed in Grenoble, but very rapidly turned into an EMBL-wide collaboration, and is using the 3 EMBL labs doing crystallography and supported by the 3. But it is also being used in many other academic laboratories, even in industry as well. And there is quite a large user community behind CRIMS, which is an advantage for us. CRIMS has different modules. It has a module that deals with crystallization and crystal farms. It has a module that deals with communication with the synchrotrons, and it has a module that we have developed recently in collaboration with Gerard Biconi and the global facing team for automated data processing in the context of fragment screening. And I will come back at some point at a later point. And you can use these 3 modules in a completely independent manner. You may not need the 3. So how does the system works, this concept of online crystallography? Well, thanks to the combination of the automated pipelines, including automated crystal mounting and processing, and the automated data workflows and web-based web-controlled data workflows, well, people can simply ship samples to the facility through FedEx or otherwise, purified samples. Then they will use the CRIMS web interfaces to design crystallization experiments and tune parameters, choose screens, etc. Experiments will be done at the facility. I have to say that every sample at the facility, it goes through a quality control process based on thermofluor assays that we developed, and where we can estimate the crystallization likelihood, but also provide some information about the sample. So you have a publication here describing this method, and you will have all the results of the crystallization information of the crystallization experiments and the quality control experiments in real time through the web as soon as they are available. When you identify crystals or hits that are amenable to follow-up steps, you can, based on this, through CRIMS, again, through the web, optimize, design crystal optimization grids. And when crystals appear, which are suitable for data collection, you can use the interface that I showed you before, and to decide how these crystals will be harvested, and all the information will be recorded, and harvesting will happen automatically on the next possible occasion. After this, you will have interfaces that tell you where all your harvested crystals are, in which pins, in which packs, and you can connect with the synchrotron, use your back to tell the synchrotron, well, I'm sending these crystals to you, so that the CRIMS will be able to follow all the results after data collection. And at the moment, we have communication with 3 different synchrotrons, as I said before. After data collection, if you want to do so, CRIMS can automatically copy the data back into your facility. And if you have data processing set up, then you could also eventually do the data processing there. This is, for example, an example of crystals that have been at one of the synchrotrons, and in CRIMS, after this data collection has happened, you can automatically see the crystal, how was it in the tray, the pin, and here you have the data coming from the synchrotron, and here you can see the results of data processing at the synchrotron. And if you want, you can continue on to data processing at your local facility. Okay. So this is basically the concept of online crystallography, and well, the advantages is that it removes manual and repetitive operations. So everything is faster, more efficient. It saves your time so that you can focus on data analysis rather than on traveling, measuring samples, etc. And it's very rapid throughput for samples that require analysis of a lot of crystals is really very, very powerful. Another interesting thing is that it allows you to do a very quick feedback between crystallization and data collection, because you don't have, I mean, diffraction experiments are done very rapidly after crystals are mounted. You can have very rapid feedback and optimize based on the results of optimized crystal conditions based on the results of the diffraction, rather than to have to make decisions because you have not had time to go to the synchrotron and basically collect the data. And that can speed up the advance of your projects very importantly. Another important aspect is that because we remove all this nitty-gritty operation of handling crystals and controlling beamlines, this means that suddenly crystallography becomes more amenable for non-experts. And here you have, and that's a very important aspect. Here you have a few examples of projects where we were able to support particularly challenging projects where we are able to support users. In this case, for example, 3 structures were produced in 7 weeks, which is really a record time, or being able to handle very, very difficult samples for which we use this laser-based surgery to remove, separate good parts from a crystal from bad parts. There are many examples of this. I don't have time to go into this, but one key point here is the rapidity with which you can basically progress through the whole crystallography process. And again, this is an example of 3 young scientists coming from biochemistry lab, not actually labs with experience in structural biology, but thanks to the streamlining of all the process of getting structures, getting data sets, etc., these 3 young scientists could actually produce very insightful structures that led to very good publications. So, and they were not experts, so with a bit of our help, but they were able to progress. So this, we believe, definitely makes crystallography more amenable to those for whom crystallography is not their main activity, which we think is important. Another aspect, and this is the second pipeline, the second usage that we have, is where these pipelines can be very important, is for the rapid evaluation of small molecule target complexes. And here we have a component, an example of one pharma company who sent us a sample together with lead compounds. At day 0, we set up the crystallization trays. At day 3, the crystals were ready. They were harvested, and the next day we did data collection. So within basically 6 days, the structures were available. Chemists in this companies evaluated this structure, and a few weeks later, they send new compounds. And again, in about 5 days, even faster this time, they have new structures, and so on and so on. So these are pipelines that can be used in support of medicinal chemistry and can produce very, very rapid evaluation of small molecule target complexes. And another important applications of these pipelines, and as I've said in the introductory slide, is to support large-scale fragment screening. As you know, academic scientists are more and more interested in applying structure-guided drug design to their own projects, not to develop drugs, but actually to develop chemical probes that can be used both to investigate protein function in cellular systems, but also to establish proof of concept for the therapeutic potential of a certain target. However, these were traditionally limited by the fact that these typically required a lot of resources and that most academic labs could not afford. But actually, CRIMS also enables to do automated soaking experiments through the process that you see here. We drill a small aperture in the film with laser so that we have access to the crystallization drop. Then we can automatically deliver a small amount of solution containing a ligand, for example, or any other chemical compound on top of this aperture. This solution will enter in contact with the crystallization solution through the aperture, and whatever is in that solution will diffuse into the experiment. And after a certain incubation time, we will harvest the crystals in exactly the same process that we have shown you before. And then when we obtain the structures, we will see whether the crystals, whether the small molecules are bound or not. And this is a proof of concept that we establish, with which we establish the technology. We did this in collaboration with Magali Mathieu from Sanofi, and you see how we can obtain very high resolution and very accurate description of the molecular binding of these molecules. Interesting things is that we can use solutions with 100% DMSO, so very often, so we don't need to dilute them in crystallization solutions and so on. And we can use 4 to 5 times higher ligand concentrations as you would do in normal traditional experiments that in the context of fragment screening could be quite important. And this is because of the way the CrystalDirect technology achieves this automated ligand soaking, but I don't have the time to go into the details now. Now, with this technology, we can very easily support large-scale fragment screening, supporting 500, 1,000 or even more, supporting a screening of very, very large libraries of 1,000 components, small molecules or more per project. But before we were able to do that, we had to revise our old CRIMS infrastructure, and we have to put in place a system that not only recovers the data from the synchrotron automatically, but that can also apply automated data processing. And this is something that we did with the collaboration with the Global Facing Group that has developed the PyDream pipeline. And thanks to this, we were able to establish a system where CRIMS will automatically process every dataset through this pipeline and follow the results of this dataset. So after data collection, you can send your crystals for data collection, and the day after, you can come to the lab and look in CRIMS, and here in a single interface, you will have all the results of data processing. So you can very rapidly evaluate about out of 100, 1,000 data sets, you can very rapidly evaluate those data sets which are more likely to contain ligands and prioritize them for further refinement. And you have here a series of tools in CRIMS that allow you to see the fittings of the small molecules, and also you can open these structures in code and work with them manually and so on. So this is a very efficient way to not just produce fragment screening, support fragment screening projects, but support data analysis, which is very important. This is an example of a project where for fragment screening that we supported. I don't have the time to go into the details, but this was a pre-competitive project done in collaboration with Wolfgang Janke from Novartis. And here we were able to identify different fragment hits binding at different parts, at different sites of this very important enzyme for trypanosomas brucei, that all these different sites and fragments could be used starting point for potentially for the development of therapeutics against some types of trypanosomiasis and other antiparasitic infections. This is just one example, but since then we have supported many different fragment screening projects at the facility, and these are just a few examples. And this is now a standard service in the facility. So if you're interested, it's something you can basically apply through iNEXT. I will give you a bit more detail later. So this is, again, I come to the initial slide, the take-home message. So at the facility, we are supporting 3 different pipelines, all available through iNEXT-Discovery. A protein-to-structure pipeline that can be used by crystallographers or by biochemists or people with less experience in crystallography, and can help you advance very rapidly from pure sample to data sets or even structure. A pipeline for the rapid characterization of small molecule protein targets, and a pipeline for large-scale fragment screening where we can screen libraries of 1,000 fragments or more. If you are interested in any of these, access to any of these pipelines, I recommend you to contact us to have more information, to see whether your project fits with which pipeline. And we can also help you or give you guidance on how to prepare the your application for iNEXT. You can nevertheless apply as well independently through iNEXT, so that's contacting is not a prerequisite, but it's of course something that we strongly advise. And here you have the iNEXT contact information. Now, we're still developing new possibilities of the CrystalDirect technology, and I don't have the time to go into the detail now, but recently, in collaboration with the group of Sebastian Grener and with Simon Basu from EMBL, we have been able to develop a method to solve membrane protein structures from crystals grows in lipidic cubic phase, LCP, using serial crystallography with CrystalDirect in a very rapid and automated manner. Manipulation of crystals of LCP, crystals grown in LCP is particularly difficult. Here we have been able to use the CrystalDirect technology and automated data collection, serial data collection, to actually produce very rapid structure solution for membrane proteins. And we hope that this will become another service in the facility soon. Finally, I want to say that all the technologies that I have shown are fully available. The CrystalDirect technology is the CrystalDirect harvester is distributed by iNEXT, which is a company in Grenoble. The CrystalDirect plates, these film plates that, by the way, are also very good for in situ experiments, in situ data collection, are distributed by Mitogen. And all the services that I have mentioned are also available to industry. We have recently created a startup in Grenoble, which is called Apex, which is dedicated to provide services to industry based on the CrystalDirect technology. So if you are from industry and you are not ready to publish and want to still access the services, this is what you have to do. So finally, I would like to thank the HTX Lab team, especially Irina Cornasiu, which, as I say, is a staff scientist in the team who actually was the major driving force behind all this project. Guillaume Hoffman, Anne-Sophie Hum, and Florine Dupeux are technicians who are operating the pipelines and with whom you will be in contact through the CRIM system about your samples very often. Peter Murphy and Raphael Bourgeois are developers, and Raila Raimova is a postdoc who is working on some research project, but also on developing further possibilities of the CRIM pipeline. Just finish by saying that currently we have postdoctoral opportunities open in the lab, and if you are interested, you are welcome to join us. And this is both for the wet lab technologies as well as for the software technologies as well. So with that, I would like to finish. Thank you for your attention, and I would be happy to answer your questions if you have any.